It's great to have you all here today, tuned in for our OzFlux Ecosystem Exchange webinar series. Please feel free throughout the talk today to enter questions through the webcast using the Q&A function found at the bottom of your screens there, and then we'll answer them towards the end or after the two speakers today. If you have any technical issues, please feel free to comment in the chat function, and we'll have our technical support team address those as needed. And so thank you all again for being here today. It's great to see a huge number of participants from a wide range of organisations around the country and around the globe, including New Zealand, South Africa, China and Indonesia, to name a few that we noticed in the sign-up sheet. So welcome to all of you. So to start today off, and in keeping with the spirit of reconciliation, when my screen changes, apologies here, my screen is not moving for me, unless it is for you. There it goes. Okay. Hopefully you can all see my acknowledgement of country screen. I apologise for some technical difficulties of my own there. And so to start today, in keeping with the spirit of reconciliation, we would like to acknowledge the traditional owners and custodians throughout Australia and New Zealand and all nations. We honour their profound connections to the land, to water, to biodiversity and culture, and pay our respects to their elders, past, present and emerging, as well as acknowledging the important role that the Aboriginal and Torres Strait Island people continue to play within our community. I myself am joining you today from the lands of the Noongar people, who remain the spiritual and cultural custodians of their land in southwest WA, and continue to practice their values, languages, beliefs and knowledge. And so it would be great throughout the day, throughout the talk today, if you could let us know where you're tuning in from in the chat area as well. And so for those of you, another quick question, acknowledgement as well to our OzFlux partners, and we're supported by TERN through the NCRIS facility, and we also contribute to the FluxNet global network, and we have a variety of public organisations in Australia and New Zealand. And so today, my name's Caitlin Moore, for those of you who haven't met me yet. I'm the OzFlux Communications Officer, and I am also a lecturer at the University of Western Australia. And it's my absolute privilege today to introduce to you our two speakers, Professor Elise Pendall and Dr. Peter Isaac. And so they're going to be talking today about estimating ecosystem respiration from flux tower and chamber data, giving us some insights from the Cumberland Plain site. Professor Elise Pendall leads the soil biology and genomics theme at the Hawkesbury Institute for the Environment at Western Sydney University, where she works really closely with postdocs and students to conduct basic and applied research in ecosystems in Australia and beyond, including the Cumberland Plains Woodland TERN OzFlux site that I've just mentioned. And Dr. Peter Isaac is the data coordinator for TERN Ecosystem Processes node and coordinates data at the Cumberland Plain site. He helps people make sense of their data and has visited most of the OzFlux sites and has found it a privilege to work with such a dedicated and competent group of people. And so without further ado, I would like to now pass over to Dr. Peter Isaac, who will be leading us in the talks today, who will then be followed by Elise Pendall. Peter, over to you, and I will stop sharing my screen.
Thank you, Caitlin. Is my audio working and my screen shared?
I can see your screen and I can hear you, so I will stop sharing. Well, I'll stop showing my video.
Okay, thank you for those kind words, Caitlin. I am speaking today from the lands of the Wurundjeri people, and I wish to acknowledge them as traditional owners of the land I'm on today. I would also like to pay my respects to their elders past, present, and Aboriginal elders of other communities who may be here today. The title, Estimating Ecosystem Respiration from Flux Tower Data, Insights from Cumberland Plain. Introduction. A question that's intrigued me for a long time now is how good is our estimation of ecosystem respiration from flux tower data? Now, that's a huge question, and we're not going to cover all of that today. The talk that I'm about to give is a small contribution. Elise's talk up second is a much larger contribution to the science, so stay around for that. But we're just going to nibble at one part of this. It's a much bigger question, and there's a lot more work to do on this problem. This talk, my talk, will cover how do we know that the CO2 flux from Cumberland Plain is noisy, as number one. Secondly, what effect does that have on our estimates of ecosystem respiration? And thirdly, what can we say and what can't we say about ecosystem respiration, gross primary productivity, and net ecosystem exchange from flux tower data? Going to the first one, how do we know the CO2 flux data from Cumberland Plain is noisy? And I'm presenting here some work that Alison Bennett has done and has published recently in Global Change Biology, and I'd refer you all to her paper. It's well worth reading. The part of this plot, what she did was she plotted out gross primary productivity from a range of the OzFlux towers and looked at how they vary with temperature. And that's the main plot you can see on the left-hand side here. Now, when she did that, she noticed that a few of the sites had quite a bit of scatter in the GPP data as a function of temperature, in particularly Cumberland Plain. And so I'm circling that with my mouse now. It's, if you can't see that, it's the top right-hand plot in that large range of plots there. So the top right-hand one, that's GPP from Cumberland Plain as a function of temperature. And you can see that it's quite noisy. It spans a very large range compared to the other sites. And I've done a blow-up of that on the right-hand side to show a bit more detail. And in that, I've highlighted a couple of things. Alison went through and plotted everything from the 50th percentile up to the 99th percentile. They're the lines that you can see. And what she found at Cumberland Plains was that around about the 95th or 92nd percentile, we started seeing peaks in that GPP versus temperature response that were well off the optimum seen at the other percentiles. And the conclusion was that this change in the optimum temperature at Cumberland Plain was an artifact of the noise in the data. So that triggered off a cascade of things. Then Elise and Dan Metzen, in particular from Cumberland Plain, got in touch with me to say, what do we know about Cumberland Plain? What can we say about the data there? How can we reduce the amount of noise and then improve Alison's analysis? As always, before we got into this, we asked a simple question. Is this a sensible thing to do? So is what Alison's trying to do here a sensible thing to do? And what she's really looking to do there is to look at the sensitivity of gross primary productivity to temperature. Now, it turns out that that happens to be a very sensible thing to do and something quite close to my heart for a long time back now. The first reason that it's really important is because that sensitivity to temperature is really going to determine whether we've got a positive or a negative feedback to a warming climate with GPP. The second thing is land surface model validation. Now, in CABLE, which is the Australian land surface model, the temperature variability and assimilation goes all the way back to a couple of papers by Kalatz in 91 and 92, and you can trace this all the way back through Ray's papers in the early days of CABLE. And in particular to Kalatz's 1992 paper, and I've got a little plot there on the right-hand side that comes straight out of that paper, which shows the variation of assimilation with temperature for both C4 and C3 vegetation. Now, there's not a lot of evidence for that at either the leaf scale or particularly the ecosystem scale level. So anything that we can do there to try and validate those temperature responses is a very strong contribution then to the faith that we would have in land surface models. So that makes it a very sensible thing to do. Unfortunately, flux towers measure net ecosystem exchange, not gross primary productivity. We get gross primary productivity by partitioning net ecosystem exchange into GPP and ecosystem respiration. But to get GPP, we need ecosystem respiration. We don't measure that. We have to derive it in some way from the flux tower data. I'll show later on in this talk that ecosystem respiration is mostly, about more than 80% at most sites, mostly modeled as a function of temperature. Is there a circularity then in what we're trying to do? We have a quantity involved in our analysis that's modeled as a function of temperature, and we're trying to use a derived product from that to work out a response to temperature. Well, there is a bit of circularity there. We minimize that circularity though by using a neural network to model ecosystem respiration. And the neural network has no prior knowledge of any temperature function. It simply gives you back what it can find in the data. Now, the circularity that's still present is that we're feeding temperature as an input to the neural network. So we are constraining the drivers that it's got. But in our case, the neural network that we're using does a PCA up front, and it's going to discard anything that doesn't contribute a certain amount to the variance in the predictions. So there is a bit of circularity, but we're hopeful that we've minimized that by using a neural network. Just to recap on how we get to gross primary productivity, we measure net ecosystem exchange using eddy covariance and profile systems on the towers across OzFlux. We take out bad data due to all sorts of reasons, huge number of reasons. We calculate net ecosystem exchange as the sum of the turbulent flux of CO2 measured by the eddy covariance gear and the storage term measured by the profile. We then estimate a U star threshold, and I'll use that U star subscript TH from here on as a shorthand for the U star threshold. We estimate that threshold from nighttime data. So at nighttime, GPP is 0. Whatever we measure should be ecosystem respiration. We then train an ecosystem model on that nocturnal U star filtered net ecosystem exchange. So we have to have a model to try and fill the gaps that will be in that, particularly during daytime. We have to train that model on our nocturnal U star filtered net ecosystem exchange. Then we're going to apply that ecosystem respiration model to the full data set. And I'll speak a bit more about that later on. What's the hypothesis for noisy GPP at Cumberland Plain? Well, noisy CO2 flux measurements would be the first one. And then having those noisy CO2 fluxes feed into noisy net ecosystem exchange. Eddy covariance is an inherently noisy measurement. We know that. So we expect some level of noise. Are we seeing more than we expect at Cumberland Plain? The next possible source under CO2 flux measurements is the storage term. We're getting that from the difference of two large numbers, and that in itself leads to a magnification of the errors. So two things going on there. Eddy covariance is noisy, and storage is the difference of two large numbers. The next thing is that we could have a poorly defined U star threshold. Now we use that U star threshold to guarantee or to try and guarantee that we have well mixed conditions during the measurement. If we've got a poorly defined U star threshold, then we may well be violating that well mixed assumption. And lastly is site characteristics. Now I'll spend a little bit of time now talking about the site characteristics. In the top left hand of this slide is a zoomed out image from Google Earth showing the location of Cumberland Plain with respect to Greater Sydney. So Cumberland Plain sits in the top left or northwestern corner of the Sydney Basin. And it's surrounded by some fairly significant terrain, still under native vegetation here, which ranges from 700 or 800 meters high to the west of Cumberland Plain, round to about 400 meters high to the north of Cumberland Plain. So several things are coming into play there. One is we've got significant terrain to the west and the north of Cumberland Plain. We may expect some kind of drainage flow during the night. Secondly, that Sydney Basin itself opens out to the sea in that southeastern corner. So we may expect some kind of sea breeze effects during the day. Both of those things are brought home in the plot. Oh, I beg your pardon, it's gone forward one slide. The plot in the top right-hand corner, which is a fingerprint of the wind direction and wind speed. Looking at the wind direction, we can see, based on the colors there, that the wind direction is mainly westerly through to northwest at night, and mainly easterly and southeasterlies during the day. And that day-night switch in directions is consistent with drainage flow at night and sea breeze during the day. So we've got some mesoscale flows here, might lead to some kind of advection. The other thing about the Cumberland Plain site is it's situated in a piece of remnant native vegetation. It's relatively small. The dimensions there are about 850 meters across the narrowest dimension where the tower is, and about 1.5, 1.6 kilometers long. When we go through and plot footprints, and this is some work done by Cecilia for ecosystem processes, when we plot footprints for the site, these are from just a collection of days in June, we can see that at times the footprint of the tower starts to extend outside that patch of native vegetation. That happens mainly at night during light wind and stable conditions. Yes, I acknowledge there might be some problems there with some of the formulations for footprints in those conditions, but there's a heads up there that sometimes the tower footprint might be extending to the borders or just outside of that patch of native vegetation. So based on this location, we may have some advection, and we may have some footprint issues as well based on the size of the vegetation patch the tower is located in. How do we know that the data is noisy? Well, the main plot there with all the blue dots is a plot of the raw CO2 flux time series from about 2013 through to 2020 when I did all of this work. And certainly from 2014 onwards, it looks pretty noisy. There's a large spread of dots there. Why might that be important? Well, it turns out that our routines for fitting or for detecting a U star threshold are statistical routines. They're pretty sensitive to outlier points. So the outliers are going to upset our U star threshold detection. And when we get to using neural networks to gap fill data, they're very sensitive to noisy data as well. And so those outliers will be treated by the neural network as true data points, and that will affect the quality of the predictions. All up, we've got 77% of the CO2 flux data is available after quality control. That breaks down to about 38% daytime and 37% nighttime. So I just want to reiterate that nighttime value. We're down to about 30% of the available data is at nighttime. The bottom right-hand plot there is a histogram of U star threshold by year from the 4 methods that are available in PyFlux Pro. We've got 3 methods that use the change point detection algorithm from Alan Barr's paper, and then the moving point threshold from, I think, originally Marcus Reichstein. And what we note about this plot is that there is not a consistent pattern across all years of U star threshold. So using this quite noisy data, we don't get a U star threshold for many years from many of these methods. The U star thresholds that we do get have a great deal of variability between methods and from year to year. What did we do first of all? The first thing we did was we went through and applied some additional quality control to the data. You can see in that main plot there on the left-hand side, this is now a much better looking data set. It's much more constrained. You can see in the top left-hand corner there, that's the original data set. The cleaned up one was much better, much more constrained. The big addition to that was the use of a diurnal check. I can describe that a bit later on if people are interested, but it's a pretty tight filter. I don't like using it because it's got the ability to throw out a large amount of data. I don't think it should be used if you're going to be pushing your data through to any kind of process-based study. I justify its use here on the basis that we've got some statistical processes downstream that are sensitive to outliers, and I wanted to clean up the data set and see if that would then work with those statistical processes. It's a dangerous one to use. In going through and using that tighter QC, we dropped down now to 67% of data availability, and of that 33% is available at night. So we've thrown out a few more % of data. We're gradually reducing the amount of good data that we've got available to us for further analysis. Now, again, bottom right there, a plot of U star threshold from the various methods. We're doing slightly better. We've got a U star threshold for a few more years, but not all of them. And if we look at that plot again, there's not a great deal of consistency, but it's suggesting a U star threshold of somewhere around 0.2 to 0.25 meters per second. Still a very noisy number though. That threshold is not well defined from those techniques. Does it work at other sites? So do our thresholding techniques work at other sites? And so just to prove that they do, a plot on the left-hand side from Calparim, we get a value of the threshold from 92% of the available years across all methods. We can see most of the methods give us a U-star threshold in most years. There is a bit of variability between the methods, particularly between MPT and the CPD techniques. There is a hint of change after the fire in 2014. Not sure if that's statistically significant, but at least I can explain why it starts to degrade there or would decrease there. If we move on to the RU site, this site at least had profile measurements prior to 2017. And again, we get a U-star threshold for about 75% of the years available. Again, a difference between the different types of techniques used to get them. But again, we get reasonably consistent answers. So we know that it works at other sites. What else might be available to us at Cumberland Plain? And I acknowledge here there's an amazing amount of work that Dan Metzen put into this. So Dan went through and started plotting out spectra and cospectra for vertical velocity and CO2 for nighttime conditions. And we were particularly interested in looking at, do we see a threshold in friction velocity as a proxy for turbulence when these spectra and cospectra start to behave themselves? Is there a definite threshold there where above that value the cospectra look good and below that value the cospectra don't look good? And Dan did an enormous amount of work running EdiPro, getting the spectra and cospectra out, and then plotting those up. A huge amount of work, and in the end, it was not particularly conclusive. The results of that spectra and cospectra analysis, can we use cospectra to select turbulent periods at night? And the answer is possibly. But if we do, it's very labor intensive. Now, if you go all the way back to the start of boundary layer meteorology in say the 50s and the 60s, this is exactly what they used to do. They used to select runs on the basis of spectra and cospectra and discard everything else. And that's basically what we'd be getting back to. You'd end up with extremely small number of points remaining, but you'd have faith that they really were turbulent transport. But it's labor intensive. What we did get that was more useful to us immediately was that just using the EdiPro flag equals 0 is no guarantee of well-mixed conditions or good data. So even using EP flag equals 0, it's no guarantee that you're going to get a good measurement of CO2 flux at night. Looking at the spectra and cospectra at around about 0.25 and just above that, what we think is the threshold, again, no guarantee that we're getting well-mixed conditions because the cospectra for those friction velocities still look very noisy. What we did find was that the cospectra start to show some decrease at low frequency, and you can see that in the bottom right-hand corner plot here for friction velocities of 0.45 meters per second and above. We at least start to see some tailing off in the cospectra at low frequency. We don't see that for friction velocities below 0.4. And that's some, I guess, some suggestion there that the low frequency part of the cospectra starts to go to 0 once we get up to higher friction velocities, which is what we'd expect or hope to see. Below that, there's a lot of low frequency signals still there. Maybe there are some bigger processes going on that we're not capturing. So, does a U-star threshold exist at Cumberland Plain? The two top right histograms are the ones you've seen before for Cumberland Plain on the left-hand side and RU on the right-hand side. The two main plots on the screen are for Cumberland Plain and RU. The plot on the right is from Ian's paper in Biogeosciences 2016, well worth reading, looking at storage and U-star thresholds, etc. at RU. On the plot, we show turbulent flux and storage component as a function of friction velocity. So the data is binned according to friction velocity, and then we take the average over those bins. For Cumberland Plain, the turbulent flux is the circles. And we can see that the turbulent flux, CO2 flux, rises as CO2 increases, but it doesn't start to flatten off until we get above about 0.6 meters per second. So that's kind of suggesting that the U-star threshold at Cumberland Plain is right up around about 0.6 meters per second. The similar data at RU, which is the gray squares here, that starts to taper off at around about 0.4 meters per second, which is consistent with the numbers on that histogram, where we're seeing a U star threshold from the CBD and the technique at least at around about 0.4 meters per second. Interestingly enough, in the plot for Roo, the storage term from the profile system drops to 0 at a friction velocity of around about 0.4 meters per second. Entirely independent set of measurements, bang, it's dropping to 0 at about 0.4 meters per second. That's pretty good evidence that the U star threshold at Roo is around about 0.4 meters per second. Cumberland Plain, the storage term is done in the open squares, and it basically never drops to 0. It gets down to about 0.5 micromoles per meter squared per second, but it never drops to 0, not even when we get up to friction velocities of around 0.6. So Cumberland Plain storage remains really large, non-zero, at large friction velocities, and the turbulent flux becomes constant more or less once U star gets above 0.6. These would suggest that the U star threshold is not well defined at Cumberland Plain, and if it is there, it's going to be at about 0.6 or greater. What happens when we start looking at just the noise of the data? What else? What other evidence do we have that it's noisy? These are uncertainty plots produced by Ian McHugh. The top right-hand one is for Roo, and again, that's from his 2016 paper. And what we're plotting here is an estimate of the random error as a function of the flux magnitude itself. So the lines here are estimates of random error plotted against net ecosystem exchange. In the case of Roo and the plot beneath it, which is Boyajian, for both of these sites, we get an intercept of this random uncertainty at around 2 to 3 micromoles per meter squared per second. And if you go through Ian's paper and also through Richardson and Hollinger, where the method came from, there are good arguments why we would expect to see a bottom limit of around 2 micromoles per meter squared per second. So both Roo and Boyajian are giving us intercepts in the range that we expect. For Cumberland Plain, when we go through the same analysis, we're now getting an intercept here at above 4 micromoles per meter squared per second, which is considerably higher than we expect to see. So there is some evidence there from an independent or objective method that the Cumberland Plain CO2 flux data is just noisy. CO2 flux and friction velocity at Cumberland Plain. We know now that the CO2 flux at Cumberland Plain is noisy. Better QC has helped, but at the expense of throwing out some data. And an uncertainty analysis shows we've still got a fair bit of noise in that data. Threshold detection is not conclusive. We get different and inconsistent values from different methods. The value that we get differs from a simple CO2 flux versus friction velocity, and the storage term doesn't drop to 0 at high friction velocity. Where do we go? We want to make some progress. Where do we go? So the next question to ask really is, how sensitive are net ecosystem exchange, GPP, and ecosystem respiration to the choice of friction velocity threshold? Just to recap on the type of U-star filtering methods that we've got available in PyFlux Pro, there's a basic method which just removes nighttime data when the friction velocity is below the threshold. All daytime data is accepted. EVGB, Eva van Gorsel's method B, as an alternative to her method A, which was published, I think, back about 2007 in the TELUS paper. Eva's method B removes all CO2 flux at nighttime once the friction velocity falls below the threshold after sunset. And then even if the friction velocity rises above the threshold after sunset, that data will still be rejected. So it's only accepted after sunset as long as the U star is above the threshold. Once the U star falls below the threshold, the rest of the night is rejected. All daytime data gets accepted. FluxNet is the method used by FluxNet in the 2015 synthesis. That takes out CO2 flux for this period and the following period whenever the friction velocity falls below the threshold. And it does that for both daytime and nighttime. And the fourth method that we've implemented in PyFlux Pro is the same as FluxNet, but all daytime data gets accepted. There's some discussion there about whether we should be applying this, this kind of filter to the daytime data. So what does the availability of our ecosystem respiration estimates from the flux tower look like as a function of friction velocity? Plot on the left-hand side is a histogram of Cumberland Plain for the 4 different methods: basic, Eva van Gorsel's method B, the FluxNet, and the FluxNet plus daytime. as we steadily increase the U star threshold. And as you can see, when you've got a threshold of around about 0.1 meters per second, we get somewhere between 15 and 27 or so % of the data is still available after applying the filter. By the time you get to a U star threshold of about 0.5, we're down now to 2 or 3% of the data. The amount of data that's rejected depends on the method. Basic accepts most of the data. The two FluxNet methods accept the next highest amount, and EVA's method B is quite a tight filter. It throws out the most data. In terms of the sites across the OzFlux network, this is the plot on the right-hand side. You can see that in general, once we've applied the U star filter for all of these sites, we range between about 7 or 8% of available data. I think that's even Cumberland Plain, all the way up to around about 22, 23% of the observations available from some other sites. So there's a fairly big range, but they're all quite small. If I was to replot that in a different way, so now this is a histogram that goes up to 100% to indicate all of the data. The blue is the observations and the red is the modeled data that we are going to backfill using our eco respiration model. This plot now shows the extent to which we're relying on the ecosystem respiration of the tower. The majority of our data, 80% at most sites or greater, is going to be derived from some kind of model of ecosystem respiration. That's a lot of data being filled from a model. What's the sensitivity to the U star threshold value and to the type of filter? These plots are time series of the monthly total of ecosystem respiration at the top, gross primary productivity in the center, and net ecosystem exchange at the bottom, as a function of year on the x-axis, and in the left-hand plot as a function of the friction velocity threshold for the basic U star method. We get some separation based on the threshold chosen in the 2013, but for the rest of the time, most of those lines overlie each other. There's not a lot of sensitivity in the monthly totals to the choice of the U star value. If we move to the right-hand plot, this is now choosing a threshold of 0.3 meters per second, somewhat above what our CPD and MPT techniques told us. But we're now applying the 4 different types of filter available to us. And again, for these monthly totals, we don't see a great deal of sensitivity to the threshold filter that we're applying. So in general, the monthly totals look like they're reasonably insensitive or somewhat insensitive to both the choice of threshold value and the type of filter used. If we look at the cumulative, and this is now starting to get to be a different story. So now I'm plotting out the cumulative carbon budget quantities across that time series of data from 2012 up to current 2021. The top one, ecosystem respiration, middle panel is GPP, and the bottom one, which has got a bit more information in it, is net ecosystem exchange. On the left-hand side, again, we're looking at the different values of the U star threshold. And we can see that it starts to separate out quite a lot by the time we get to the end of the data stream. In fact, there's around about 7 tons of carbon per hectare per year range in looking at all of those U star thresholds. So there's quite a big range. In terms of the different methods, the range is not nearly as much. Again, the cumulative totals not quite as sensitive to the choice of filtering method as they are to the choice of threshold. What does that mean? If we're looking at around about 7 or 8 tons of carbon per hectare per year uncertainty at the end of an 8.5 year long data set, then maybe our uncertainty per year is something around a ton of carbon per hectare per year. We might be better than that because I'm using a very wide range of thresholds here. We might be able to narrow that down to a narrow range of thresholds and get a better uncertainty. But just as a ballpark figure, somewhere between 0.5 ton and a ton of carbon per hectare per year might be the best we can say in terms of uncertainties for carbon budgets. Conclusions: CO2 flux at Cumberland Plain is noisy. We need to do a bit more work to find what the reason for that is. The threshold at Cumberland Plain is not well defined. And we really need to do some further work here to figure out what's the best filter method, what's the best value of the threshold. It turns out that seasonal CO2 budgets, like monthly totals, are relatively insensitive to the U* threshold. And that's good for doing ecosystem process studies. What's not quite so good is it turns out that the cumulative CO budget is quite sensitive to the U* threshold and the filtering technique that we use, and that may set limits to the uncertainty of long-term budgets derived from flux tower data. And that's it from me. I'll stop sharing my screen. How do I do that? I think I did.
Okay, so thank you very much, Peter. That was perhaps a little bit discouraging, but certainly enlightening. And yeah, so it's great to have an opportunity to follow up on that excellent analysis of the eddy flux data. First, I want to acknowledge the traditional owners of the lands that I've been living and working on since around 2014, the Darug peoples, and pay my respects to the elders past, present, and emerging. So this presentation that I'll give today has resulted in part from an ARC grant that I got with Vanessa Havard before she passed away. May she rest in peace. Also Eric Davidson, Mark Cholker, and Stefan Arndt. And so, and really what I'm going to share with you, it's a big team effort. There've been a lot of people involved in this. And I especially want to acknowledge our postdoc Namjin No. And how do I make this go away? And he's over here. And our postdoc Namjin No has recently moved back to South Korea to take a faculty position. Alexei Ranchon did a lot of work on soil and ecosystem respiration for his PhD. And we also had a visiting scientist, a student, Jinchuan Li, who is involved in a lot of the field work, which when we're doing component measurements from chambers, it's quite labor intensive. And so that's mainly what I'm going to focus on today. So as we've just heard, ecosystem respiration measurements can be quite fraught. Nighttime eddy flux measurements are challenging at most sites, not just at Cumberland Plain. And as we've seen, the resulting values depend on which quality control methods we might be applying, and especially the U* threshold method. Of course, our accurate estimation of the carbon balance depends on accounting for storage at most sites. And I think that a lot of our noise is attributed to the challenges of accurately measuring the storage component at our site. And despite how careful we're being in measuring and accounting for the nighttime storage and screening out the noise from the turbulent fluxes, I think we still often can see elusive and non-quantified advective losses and gains that are contributing noise. And so this is where I'm hoping that component measurements of respiration can help. So I'll be asking the questions, is scaling up of respiration components in time and space adequate for validating our ecosystem fluxes measured from eddy covariance? And can these component fluxes provide any mechanistic insights that can help inform model formulations? And so just as a little overview, I've got the canopy carbon balance shown here. And I'm going to be focusing today on the ecosystem respiration, which is the sum of leaf, stem, root, the autotrophic components, and then also microbial respiration from the soil and litter respiration. I would like to also point out that in cable pop, which is Vanessa Havard's branch of the cable model, there are several functions that are used to calculate these respiration fluxes. The autotrophic components, leaf, stem, and root, they're mainly a function of temperature. And right now, the way the model is set up is that the maintenance respiration is calculated for these components, whereas growth respiration is calculated for the autotrophic system in all in one as one. And then the microbial respiration, or the also known as decomposition, that's calculated as a function of soil temperature and water, soil water content. And we've also added substrate availability, the carbon substrate availability that might also be partially limiting or regulating the microbial respiration. Excuse me. And so as part of the ARC grant, Vanessa was able to build a component or an option using the dual Arrhenius Michaelis-Menten or DAM model for soil respiration. And that's been incorporated now into cable pop. And just really quickly, the Arrhenius portion of the model, we can think of that as the maximum velocity of the enzyme, any enzyme reaction. In this case, it's decomposition of organic matter, where the substrate concentrations aren't limiting. The alpha CO2 is a pre-exponential parameter. We can think of that as the enzyme capacity. And then EA is the temperature sensitivity. The Michaelis-Menten component of this respiration or decomposition function, we have mainly, the main thing that we are looking at is the concentrations of the substrate or the soluble carbon, and that's multiplied by a diffusivity, and then also the soil moisture content to the third power. And oxygen, the lack of oxygen actually can limit decomposition rates, and so that's also incorporated into the DAM model. So we've made a whole lot of component measurements. Some are continuous and some are campaign-based. I've got some pictures of these. So we have a number of different types of instruments measuring the stem and soil respiration, the microbial respiration, the root respiration and litter. These components are mainly by difference from root exclusion or litter exclusion, but we also have some detached root respiration measurements that were there was kind of some separate experiments done on those, and I'll share a little bit of that data with you later. But in addition to the fluxes, of course, we have to have some ancillary measurements to allow us to do appropriate scaling or gap filling and modeling. And so, of course, we use soil, air, and stem temperatures, soil water content, soil microbial biomass, and then the surface area of the stems, all the tree trunks per meter squared of ground area. And in some cases, we might be using root or litter biomass for our scaling. So here's what some of these measurements look like. Of course, we have the ecosystem respiration, and mostly what I'll be showing you is actually nighttime data, but we also have some continuous measurements that are some continuous estimates that were partitioned using the neural network, as Peter was describing before in PyFlux Pro. Here's an example of some of our soil respiration measurements. This is an auto chamber made by Lycor. We also have done some litter exclusion, or just litter removal. And then also we have done root exclusion, where we sort of dig out a little bit of soil and take the roots out, and then put the soil back into a little section of a little kind of column where we have this natural soil, but without roots growing through it. And since we're trying to do relatively low disturbance, we have this other kind of flux chamber called the EOS FD, a forced diffusion chamber. And we have here's our little respirometers that Craig Barton helped us build to be able to measure root respiration and determine their temperature sensitivity on detached roots. We've also gotten quite a lot of data on stem respiration from a couple of different types of options here. And so I'm happy to talk in more detail about any of those measurements and the instrumentation later if anyone is interested. So earlier this year, Alexis Ranchon published a paper on looking at the temperature sensitivity and comparing ecosystem and soil respiration. And here we'll start out by comparing the parameter fits using the Lloyd and Taylor function. And so this was published in 1994. It was originally based on soil respiration data, and it's been widely used to for gap filling of ecosystem respiration, even though it was based on soil data. And it's only using temperature, soil temperature as a driver. And so here what we have is soil respiration data from 4 years fit to the Lloyd and Taylor parameter. Each of these points on the left-hand side for seasonal data, that's a monthly data point. On the right-hand side, the diurnals are each 24 hours of the day for the soil respiration. And then down here on the lower panels, it's just the 12-hour nighttime data from ecosystem respiration. And so you can see that there's some similarities between the temperature response functions on the seasonal scale, but on the diurnal scale, look a bit different. And so after we extracted the parameters from those curves, we can see that ecosystem respiration seems to have a somewhat higher temperature sensitivity than the soil respiration. But we also notice that the temperature sensitivity, the E0 value, is as dependent, or at least as dependent on the time scale of the data that we're using as on which flux we're examining. So that's just a bit of food for thought, I guess. Because, really, we know that temperature and moisture are interacting to drive soil and ecosystem respiration. They're not really, operating independently. And so also from Alexis' paper, these are plots using the DAM function of ecosystem respiration of the nighttime data only in the top left. And then we have three different auto chambers for the soil respiration. And, the first thing that you notice that there's sort of a similar shape of all of these fits. I should probably just orient you to the plots a little bit. Here's the temperature axis and the soil water axis. And then of course, respiration is the vertical axis. And we can see that there's sort of a broad optimum in the water content values at high and low values of soil moisture, the respiration rates decrease. And the temperature sensitivity is roughly exponential, but it's steepest sort of at intermediate water contents. And so I have a couple questions, not necessarily a lot of answers. One question might be, is it appropriate to use the DAM model for ecosystem respiration? This was originally developed for microbial and soil respiration. Well, Lloyd and Taylor have done it, and now we've published it. So maybe that sort of implies that it's maybe an appropriate thing to think about. Furthermore, what's the best model to use for gap filling? A parsimonious one like Lloyd and Taylor, perhaps a more mechanistic one like DAM, or where we have the appropriate data sets, perhaps neural networks have the least potential bias in them. So, and so here's some comparisons of the temperature sensitivity parameters from the DAM model between soil and ecosystem respiration. And in fact, here we see that soil respiration seems to have the higher temperature sensitivity than ecosystem respiration. Okay, so the story changes a little bit when we add the water content and use a different model to evaluate our temperature sensitivity. And again, the time scale matters. in terms of how we're averaging our data. And so, yeah, so I think it's, I'm hoping that, you know, eventually we can start using some of this empirical knowledge to, you know, potentially inform or at least challenge the models that we're running. And we'll get to that in a minute. But here, let's take a look at some nice work that that Namjin has done. We actually presented this at HEU a couple of years ago, or a year and a half ago. But so what we've done is we've taken the eddy covariance ecosystem respiration based on, it's a daily, these were daily averages. I think they're daily averages, but just reported as micromoles per meter squared per second. And that's in the solid blue line. This is for a year of data or so. And the dashed dotted blue line is the sum of soil and stem respiration from the chambers. Okay, and so to me, this is actually really encouraging that we see, you know, quite a good correspondence in the time series, very good correspondence in the temporal dynamics of the ecosystem respiration measured on the flux tower, you know, a little ways away from where the chambers were doing the measurements. And let's see, so we also have, that's the soil respiration separately. That was estimated from auto chambers and gap filled with the dam model. Microbial respiration is this yellow line, and that was from the root exclusion chambers, and also gap filled. And the root respiration, that bright green, that is obtained again by difference from the difference between the total soil respiration and the root exclusion and microbial respiration values. And here's our stem respiration measurements in the green. And Namjin has published a nice paper on that earlier this year as well. Yeah, so that's been pretty promising. And so the next steps we've taken is to evaluate how the dam model is representing the data that we've obtained. And so again, Vanessa was able to implement the dam model in cable pop. And so there's three now kind of two or three main options for how we represent heterotrophic respiration or microbial respiration in cable pop. And so let's just do some data model intercomparisons here. Well, we're also looking at, on the left-hand panel, this is the ecosystem respiration from the eddy flux on the X, and ecosystem respiration as the sum of soil and stem respiration on the Y-axis. And, you know, yeah, we might have noisy flux data from the flux tower, but this looks pretty good to me. It's quite encouraging. Here in the middle panel, we have ecosystem respiration from the model versus soil plus stem components. And now we have the model versus the eddy flux data. And so here we see, again, really good agreement, but with a bit of an offset. So to me, this is all actually very, very encouraging. And so I guess, you know, these are pretty independent measurements, although I guess I should probably point out that that the cable model has been optimized against the NEE data, the NEE and ecosystem and evapotranspiration data. So, you know, so there should be some good, you know, there should be some good agreement. Nevertheless, let's take a look at the other components now. So here we have stem respiration from the model compared to stem respiration from two different flux chambers, the LICOR and the ESFD chambers. And here, the model is only showing the maintenance respiration. So we need to still add the growth respiration back into those components. And once we do, I think that we'll have pretty good agreement in between the model and the measured component fluxes. here looking at the total soil of the, represented by the cable pop down model compared to the auto chambers. And wow, great agreement. It's not quite a one-to-one line, but we've got a really excellent R squared there. This is, actually need to check. I think this is on a year of data, but actually it might be more than that. So I think I'll need to go back and double check the time scale on these. At any rate, it's the microbial respiration that's looking kind of the best to me, because we've got very close to one-to-one relationship between how the model's doing and how the root exclusion microbial respiration measurements are going. Again, the root component from cable, we need to add back in the growth respiration component. All right, and this is one more little piece to show some of the detached root experiments. Basically, from a bunch of different campaigns, we did measured root respiration at a number of different temperatures and came up with perhaps some evidence of seasonal acclimation, which is indeed already incorporated in the cable pop model, based on a paper by Cholker and Atkin and others in the early 2000s. So let's just add it all up. And so this is just for the year from September 2018 to August of 2019. And we see quite a good agreement. The ecosystem respiration is the sum of soil and stem. We get about 1,300 grams per meter squared in a year, compared to the 1,400 from the eddy flux, and about 1,300 from cable pop down. So, with this is for ecology, this is pretty good. For noisy data, I feel like we're doing okay. However, the annual sum in so the annual sum here does include both growth and maintenance respiration, but we haven't incorporated leaf respiration into the sum, but it is of course included in the eddy flux. So even though I feel like we've done quite a good job on estimating the respiration of the different components, there's still bits and pieces that we need to either add back in or somehow account for. Take out of the dam model or the cable pop dam model for a better comparison. Again, highlighting how well the microbial measurements are comparing with the model. So just to sum up, I feel like the component measurements of respiration helped quite a bit. We validated the ecosystem respiration from the flux tower measurements as the sum of stem and soil respiration. We demonstrated similar temperature and moisture sensitivities of those two main fluxes. We've quantified the microbial contribution. It's at most of soil respiration and at least half of ecosystem respiration. We can provide some parameter values for testing and doing different sensitivity analyses to estimate component fluxes at diurnal to seasonal timescales. And ultimately, I think that these types of measurements will help improve model estimates of unobservable fluxes like GPP and daytime ecosystem respiration. So that's it from me.
Wonderful. Thanks very much, Elise. That was a really excellent talk. And you too, Peter, both very informative and interesting talks. We've had a lot of interaction going on in the comments and the chat function as well. And so if you're happy with me to kick off some questions, we're actually, sorry, a brief note, we're coming up to the close of the hour. So before you all take off, I'm just going to quickly share my screen again to give you some information on the next seminar session that's going to come up for OzFlux. But we'll also hang around for questions if you've got time to as well. And so this is our information for the next OzFlux seminar. It will be later in November, so watch out for that. Do feel free to contact us via our website, either through ozflux.org.au or tern.org.au, or you can email Peter or Elise at their email addresses provided here. There will also be at the end, when you sign out of this, a link for a very brief survey as well. So do please feel free to spend a couple of minutes entering your feedback in that survey. It's really useful and helpful for us to plan for future seminar events. And so I guess without further ado, for those of you who are interested in sticking around for questions, the first one comes from Johannes Lolbach in New Zealand. And he said, Peter, it would be really interesting to see if sigma W thresholds were more consistent than new star thresholds. Are you able to comment on that, please?
The best comment is probably an apology, because I think I've been promising to do this for Johannes for 4 or 5 years, possibly longer. It's quite possible that looking or using sigma W instead of friction velocity as a proxy for turbulence, it's quite possible that will perform at least as well, if not better. The only experience we've got directly of that in Australia was when Eva did that for Tumbarumba, and found that with sigma W, the presence of gravity waves, which come through as a low frequency signal in sigma W, that caused the sigma W measurements to be higher than expected. And so it was hard to find a threshold from that. But as a generic technique, it should be investigated. And I promise Johannes that I'll do it within the next 12 months.
There we go. Note it down as an action item.
Yeah.
So then Jason has said, Peter, amazing talk, and opens up more questions and answers in some ways. It's an incredible amount of work from TERN EP Central Node, and it's not just a data processing, but world-leading scientific academic contribution, way beyond the expected. So I think that's a very pertinent comment that reflects, I guess, how we all feel about your contributions to the OzFlux community. He then has a question to ask, can we quantify the level of noise as a metric and use that as an input to the QA/QC process? Could this then identify where there are issues at other sites?
As usual for Jason, it's a really good question. That was one of the motivating factors behind my doing this work, was to see if we could come up with some objective criteria or objective metrics for deciding which sites are good, which sites are not so good, which data is noisy, which data is of better quality. We kind of get a feel for that anecdotally by looking at a large amount of data, but it would be good to have some objective measures for that. The ability to detect a U-star threshold, I think, is one of them. Consistency of activation energy estimates when you get to looking at respiration at level 6, I think that's another one. I think the work that Ian's done in terms of implementing the Richardson and Hollinger uncertainty is a third one. I think that all of these, we should be looking at all of these across all of the OzFlux sites and trying to figure out from them which ones are doing the best job of objectively, or trying to be more objective in deciding what's a good site and what's a not so good site. Yeah.
Excellent. And so I guess that then feeds on to his next question, which is one that I actually wrote down as well. I was just curious about what your thoughts are on what is the likely cause of the noisy data. I mean, Elise sort of showed that, you know, how big of an issue is it if you've got your soil respiration and stem respiration values coming, you know, fairly close to that 1-to-1 line, especially for an ecological purposes. So what, have you got any thoughts as to what might be causing it? Is it an advection issue or something else that we're not really considering at the site?
There's at least two things here, but the first thing is to say a huge thank you to Elise and to acknowledge the amazing work that she and her group have done. After going through all this stuff myself, I was getting fairly despondent about the ability of flux towers to give us a reasonable ecosystem respiration estimate. Elise restored that faith simply with that time series that compared the chamber measurements with ecosystem respiration off the flux tower. The fact that they agree quite well, in fact surprisingly well, was just brilliant. So, and that's at a noisy site. That's at a site where we know there are noise problems. What's the cause of that noise? Don't know. Would need to do a fair bit of work to find out. Candidates would be advection of some kind, and I guess that's the one that I'm thinking most most strongly of now. Although for the life of me, I can't figure out why the storage term doesn't drop to 0 at higher friction velocities. That's a real puzzle. I've been drawing pictures in my head of internal boundary layers from the edge of the forest and where that internal boundary layer would intersect with the tower. And is there something in there that could explain it? But advection would have to be the most likely candidate. We can't rule out systematic errors either. And I guess the fact that the storage term doesn't drop to 0, we'd have to think that some kind of systematic error in the measurements might be there as well. But you know, Dan looks after the system, or Dan used to look after the system, and he's just really good. So I don't. The short answer is I don't really know. We'd need to do a lot of extra. I've tried to convince Elise to mount a CO2 sensor on a troopy and then drive round and round the patch of vegetation at night, so we can see what's upstream and what's downstream.
You don't want to do that, Elise?
I think it's a question of time and resources.
Yes.
Yeah.
Did you want to add comment to that, Elise, at all?
Yeah, no, I mean, I think that there's still more we could do. And yet we have tried to do sub canopy. We had a system, eddy flux system set up under the canopy for a few months, and the turbulence was almost always so low that we were never really able to get into to measure really any fluxes out of that. So yeah, so it's a bit of a challenging site. It's very flat, but it's also, you know,
yeah.
One other thing there, in my thinking about all of this, the next possible progression, if we had the resources available in Australia, and I don't know that we do, would be to start looking at large eddy simulation of that area. So can we get an LES down to several tens or 100 meters to start modeling the flow at night? Can we even push something like WRF down to maybe 500 meters? I don't think it'll go down to 100 meters, but can we get something on a mesoscale model level that starts to look at those flows at night? That might be a great topic for somebody's honours or PhD thesis, and it doesn't require quite as much in terms of physical resources.
That's a good point. And I guess that then gets on to, in some ways, Richard Silberstein's question about the error being only maybe 0.5 to 1 tonnes per hectare per year. But in this case, this approaches 50% of the signal, which is concerning. Any comment from either of you on that?
Do you want me to go first, Elise? Okay. Thanks, Richard, for the question. Those numbers are obviously just for Cumberland Plain. They'll be different for every other site. For sites where we get a well-defined U-star threshold, such as I gave examples of Calpra Monroe, for sites where we get a well-defined activation energy from the Lloyd-Taylor method, then we might expect those numbers to be quite a bit less. So I think the answer is that we have to do this at all of the other sites. It should be part of our routine analysis. And I guess the challenge there is to find the time to build what we've got as standalone scripts and standalone methods now into PyFlux Pro so that more people can use it. But really, the site PIs, if possible, and if not them, then central nodes should be doing this for all of the OzFlux sites. Yeah.
And so there's another question here for you, Elise, from Jason. He said, first off, a comment, you've done a super amount of work using ancillary methods to cross-validate the fluxes, which is really important. We need more work like this, so congratulations. He then asks, I wonder if the noisiness of the CO2 data propagates to the magnitude of the fluxes. Do you have any further comment, apart from using DAM, on how well the fluxes compare, like with GPP, for example?
Yeah, thanks for the question, Jason. So in other words, the uncertainty would increase as along with the increasing rates or fluxes. And I think that's pretty common. Yeah, I guess I'm not quite sure how to think about it other than, ecologically. Often we look at the coefficient of variation as the ratio of the uncertainty or the variance to the mean. And, so I guess if if the uncertainty is staying at about roughly maybe 10% of the mean, I think we're doing pretty good. Most general, indicator for most ecosystem data I've ever collected. And so I also wanted to maybe get back to Richard Silverstein. I think that if I'm doing my estimate, my calculations on the fly correctly, I think 1,300 grams per meter squared is 130 tons per hectare. Is that right? We divide by 10, right? So if I'm getting 130 tons per hectare per year of respiration, then this is, the 0.5 to 1 ton per hectare is really fairly small. Did I do that right? Or do we divide by 100? I'm not good at
sorry, I was looking at the next question. Were you talking about the grams of carbon scaling up to tons per hectare?
Right, yeah.
Divided by 10, but yeah,
right, yeah,
10, yep.
So our annual flux will be 130-ish tons per hectare, right? So if we have 10, or sorry, Peter, you said 1 ton per hectare of uncertainty, that's pretty small, right?
There's got to be a catch somewhere. I'm not that fast with mental arithmetic. I need to go through.
Yeah, I was like, I was looking at questions. Yeah.
130 sounds a lot for Cumberland Plain. That's a lot of carbon.
I think it's normal.
Okay.
So a couple more questions before we'll wrap up and let everyone get away for the afternoon. Mark Carey has asked about gravity waves. What are these and what do you mean by the gravity waves?
Yeah, I saw that in the questions. Thanks, Mark. I know it's not LIGO style gravity waves. We wouldn't have any funding worries if we could do that. No, gravity waves, I guess another term for them would be buoyancy waves. Wherever you have a change in wind speed or wind direction, and I'm using my hands here to display 2 layers of air moving in a different direction or at different wind speeds. Wherever you have that in a stable atmosphere, so an atmosphere where the temperature is increasing with height, then you will get, just by virtue of that shear, you will get some waves starting to occur. And what happens is that entire layer of air starts to pump up and down. And they've seen this quite clearly in the profile measurements of CO2, water vapor, and temperature at Tumbarumba. But you start to get that whole layer moving up and down, and it's the energy for that comes from that wind shear, and the frequency is the Brunt-Weissala frequency. So that's what we mean by a gravity wave. And the issue there is that gravity wave then gives you a vertical velocity signal, which you can pick out from the sonic anemometer of some regular wave activity. So not gravity waves from collapsing stars, but gravity/buoyancy waves driven by wind shear above a stable atmosphere. Cool,
good answer. And so lucky last for the day, since we're already 13 minutes over time. One from Choi Yong Yi, sorry. In your opinion, do you think tower nighttime NEE is underestimated or overestimated at the site?
Yeah, hi, I hope Tree is still there, because I think that where he's coming from, it's a bit early in the morning, like maybe, in New York City time. So yeah, great to get your question. Thank you very much. I think that we really need to do some better work on the leaf respiration, because what we have not done yet is to add the leaf respiration to the soil plus the stems. And I think that component is pretty small, because in our forests, we tend to have quite low leaf area index, like around 1 or 1.5, because these are very open in woodlands. Yeah, nevertheless, we should, I think that once we add that in, we'll have a little bit better, a little bit better direct comparison. So I will take that question on advisement, and hopefully we'll get that answer before we submit our paper. Thanks.
Great. So I would like to remind everyone that this talk is being recorded, and so it will be made available after the seminar is finished today, and there's a survey at the end. And On behalf of you all still tuned in, I'd like to thank Elise and Peter again for 2 really wonderful talks today. Thank you very much for tuning in.
Thanks, Caitlin.
Thanks, everyone. Bye.
Everyone.
Bye.